Kinase inhibitor. Usually, kinase is an activating signal. When the ligand binds to the receptor on cell membrane, the signal is transferred inside the cell, and it can transfer the signal through kinase cascade, eventually reaches the final target for activation. If this ligand is removed, the signal should be turned off, so this is a regulated signaling mechanism. But in cancers, somehow, maybe by mutation, the receptor is always active, or the copy number of receptor is increased by overexpression. Sometimes the negative regulation is blocked. That's why the whole signaling is activated all the time, and it can generate those signals of overexpression constitutively. In kinase signaling pathway, when the ligand binds to the receptor, and this binding event can change the conformation of this receptor. An intracellular component usually have a kinase activity, and it generates lots of phosphorylation event. And this phosphorylated motif can be the anchor for many different kinase or enzymes or adapt proteins. There are several kinds of different signaling pathways can occur. So one pathway is a PI3 kinase and AKT, another kinase, and eventually it reaches to the FOXO or mTOR. mTOR is important for growth signal. Another pathway is through RAS, RAF, and MEK ERK kinase. This pathway is involved in cell proliferation and survival. And another pathway through JAK and STAT. This pathway is involved in gene expression and immune evasion. These activating pathways are all important signaling for cancer cell survival and proliferation. In the example of EGFR, one possible way is the overexpression of a EGFR receptor, and also maybe the loss of a downregulation to suppress the signaling. Sometimes it can be partnered with a HER2, another receptor protein, which can make a heterodimerization. If this happens, then autocrine EGFR signaling can further activate these pathways, and eventually it can generate the constitutive activation. So here, three mechanisms work together to make a situation worse. Here are some strategies targeting kinase. One possibility is ATP binding site inhibitor. The other possibility is allosteric inhibitor or protein-protein interaction inhibitor or bivalent inhibitor, covalent inhibitor. Let's look at one by one. Every kinase has an ATP binding site because ATP is the cofactor for kinase. The binding pocket has alpha C helix, also activating loop, and inside the activating loop there are DFG3 peptide, the unit there. So ATP can bind in this cleft. That's why this alpha C helix and DFG conformation is important for kinase activity. There are two kinds of conformation in kinase. One is the DFG in active form, and the other one is DFG is moved out, so this is the DFG out inactive conformation. So these two conformation is in equilibrium. Now that's why ATP competitive inhibitor can be two types. One type is the binding to active site binding competitive inhibitor. The other type is bind only to the inactive state and also trap that conformation and don't let it move back to the active conformation. Here are two types of inhibitor. So type 1 inhibitor bind to the active site competitive inhibitor. And then type 2 is binding to the inactive state and trap those conformation. Now, next class is an allostatic inhibitor. In this case, the drug does not bind directly to the ATP binding site, but by binding to the remote site, it changes the conformation of ATP binding site, and ATP cannot bind to the kinase anymore. So this is kind of an indirect binding, but inactivating kinase. These are the examples for the allostatic inhibitors. Next class is the protein-protein interaction inhibitors. For proper kinase function, kinase need to make a contact with a substrate. If a drug interferes with that interaction, maybe by binding to kinase site, or maybe to the substrate site, or in between. This type of the specific protein-protein interaction blocker can inhibit the kinase signaling. Here is an example of protein-protein interaction inhibitor. Usually this binding site has a little wide and shallow. That's why the drug molecule needs to be a little bigger and spread out the forms. Sometimes two molecules can be combined together, so it can increase the binding affinity and specificity. But potential problem is maybe the coupling of these two molecules, one and the other, may make the molecule a little too big. So the total size of the molecule combined, maybe that's not small molecule anymore. Definition of a small molecule is molecular weight is less than 500. This molecule may be more than 1000. Another type of molecule is a covalent inhibitor. If the molecule make a covalent binding to the enzyme, then it can be irreversible inhibitor. The conventional kinase inhibitors, even though they have a high affinity, there is a dynamics of on and off. But in this case, once the covalent binding is made, then it can be permanent the binding. That's why it may have a stronger effect, and at the same time, it may generate some serious side effect. So this is an example of a covalent inhibitor. Now here you can see the good electrophile which can make the covalent bonding with a cysteine motif. Let's summarize the kinase inhibitor classes. First type is the ATP binding site inhibitor. Two types. One is the active site competitive inhibitor. The other one is inactive state trapping inhibitor. And other binding site inhibitor is allostatic inhibitor and protein-protein interaction inhibitor for interfering with the kinase and substrate interaction. Sometimes you can connect two drug molecules together. It may increase the specificity, but it may have a problem for PK property. And covalent inhibitor can increase the activity, but you should be careful for the side effect.